So first off, I'm going to go over the amino acids. So amino acids, they're the basic building blocks of proteins. Each one combined in a chain will produce a specific protein. When you combine a long chain of them through peptide bonds, they form a polypeptide. And the different groups on a amino acid are, first off, the amino group, which is NH2. So that's this blue group right here on the left side. And then also the carboxyl group, the COOH group, which is on the right side over here in red. And then each amino acid will also have a hydrogen attached to it. So that's this black H at the top. And then finally, the side chain, or otherwise known as the R group, that's the group that's different in the amino acids. So depending on which R group you have, this will create a different amino acid. And you can recognize the different amino acids by the different side chains attached to it. And then this carbon in the middle that's bonded to these four groups, it is known as the alpha carbon. So in this lecture, I'm going to go over the proteinogenic amino acids. So these are the 20 amino acids in our body that are encoded by DNA and then RNA, and then finally made into proteins. They're called the proteinogenic amino acids because these are the 20 amino acids that our body uses to make proteins. There are other amino acids in the world, but they won't be covered on the MCAT. So all 20 of amino acids, out of the 20 amino acids, 19 of them are optically active except for glycine. And that's because glycine, glycine has a hydrogen as the R group. So because it has two hydrogens attached to it, it's not optically active. And then the amino acids that we're going to go over today, the 20 of them, they're all L amino acids. So in an L amino acid, the amino group, so this NH3+, is on the left side in a Fisher projection. As you can see on the right, the amino group is on the left side, and that creates a S absolute configuration in all 19 of the amino acids, in 19 of the amino acids, but the cysteine is the only one out of the 20 that has an R configuration because the R group is a CH2SH, so the sulfur actually has a higher priority and gives it a R configuration instead. If the amino acids are hydrophilic or hydrophobic. So hydrophobic amino acids, those are usually the non-polar ones. So it usually has an alkyl chain as its R group because of the large amount of carbons and hydrogens in the side chain without any polar molecules such as like oxygen, nitrogen, sulfur. Because of that, they'll be hydrophobic and they'll try to avoid water. So that's why they're usually found in the core proteins away from the aqueous environments that the proteins are in. On the other hand, hydrophilic amino acids, these are the basic amino acids with the amino group, the acidic amino acids with the carboxyl group, and the amide amino acids with the amides, that's R groups. So those are able to interact with the water and form favorable interactions such as hydrogen bonding. And they're usually found on the outside of the protein, so on the exterior where they have more access to the aqueous environment. And so the rest are usually called neutral, so they don't really prefer hydrophobic or hydrophilic environments over the other. And on the right, I've just included two charts. So the hydrophobic amino acids is this top one. You can see it's a lot of carbons and hydrogens, while the bottom ones, hydrophilic amino acids, they are usually the ones that have these polar groups, such as the amides, amino groups, and the carboxyl groups. Okay. So like I said before, the amino groups, the carboxyl groups, and the side chains of the acidic and basic amino acids, they can become protonated or deprotonated depending on the pKa of the group and also the pH of the environment. This is because amino acids are what we call amphoteric species, so they can either accept or donate protons depending on the pH of the surrounding. So like I said before, the amino groups and the carboxyl groups of every amino acid can become protonated or deprotonated, but then only the R groups of the basic or acidic amino acids can become protonated or deprotonated. And whether they're protonated or deprotonated, it depends on the pK of the group and then the surrounding pH. So when I was going over the acidic basic amino acids, I went over the pKa's of each R group, but the pK of the carboxyl group of every amino acid is around 2, and then the pK of the amino group on each amino acid is between 9 and 10. Usually they round to around 9. So in this chart, you see here the H hydrogen on top isn't affected, but the amino group has a pKa of around 9, and the carboxyl group on the right has a pKa of around 2. And then the variable side chain, the R group of the 20 amino acids, they're either unaffected if they're not basic or acidic amino acids, but if they are, then the pKa's of those groups determine if they're protonated or deprotonated, and also the pH of the environment. So how do you determine that if it's protonated or deprotonated? So if the pH of the surrounding environment is less than the pK of the functional group, then the functional group is protonated. But if the pH is greater than the pK of the functional group, then the functional group is deprotonated. And then there are special cases called Zwitterions where the amino acid has multiple charges, but the charges neutralize each other, and it results in a neutral molecule. So you can see in this diagram on the right is at high pHs, the functional groups are deprotonated. So the carboxylic acid has a negative charge, while the amino group does not. It has a neutral charge, and because of that, there's a negative charge on the amino acid. And then on the left side, you can see at low pHs, both groups are protonated. So the amino group is protonated and has a positive charge, while the carboxyl group is protonated and has a neutral charge. And because of the positive charge on the amino group, the whole amino acid is positive charge as a result. So then in between the two pKa's, you can have a Zwitterion where the amino group is positively charged because it's protonated, while the carboxyl group is deprotonated and has a negative charge. So at certain pHs, this can happen, and because of the positive charge on the amino group and the negative charge on the carboxyl group, these two cancel out, and you have a neutral charged amino acid, even though there are two different charges on the molecule. So the positive charge and the negative charge on that same amino acid cancel each other out, and you have a neutral molecule, and that's called a Zwitterion.